Hello students. Now stir tank reactors. The most commonly used bioreactors are stir tank reactors. Each reactor is a large closed cylindrical vessel made of steel. It has a curved base to facilitate the mixing of culture broth. It has following features like a central shaft, a stir plate or stir blade magnetically coupled to the shaft for stirring the broth. Okay, so now what is stir tank reactors? The most commonly most commonly used bioreactors bioreactors are stir tank reactors. Each reactor is a large closed cylindrical vessel made of steel. It has a curved base curved base base to to facilitate the the mixing of culture culture broth. It has following features. It has following features like number 1, a central shaft. Number 2, a stirrer stirrer plate or stirrer blades. Stirrer blades. Magnetically coupled to the shaft. Magnetically coupled to the shaft. For stirring the broth. For stirring the broth. Okay. Now, next is an oxygen delivery system or air inlet. Now next is an oxygen delivery system or air inlet or air inlet. Number 4, a foam control system. A foam control system. Next, temperature pH control systems and a cooling jacket. Temperature, pH control system, pH control systems and a cooling jacket. Next, sampling ports for removing small samples of culture from time to time. Sampling ports for removing small samples of culture from time to time. Okay. Now, next, is another kind of star tank reactor known as spurged. In spurged star tank reactor, the sterile air bubbles are spurged into the culture medium for aeration and stirring the culture medium. Now, next, spurged stirred tank reactor. Okay. In spurged stirred tank reactor, the sterile air bubbles sterile air bubbles are sparged into the are sparged into the culture medium for aeration and for aeration and stirring the culture medium. Stirring the culture medium. Next, downstream processing. Downstream processing. The recovery and purification of biosynthetic product from the growth medium is called downstream processing. It includes techniques like filtration, centrifugation, sedimentation, various types of chromatographic techniques, electrophoresis, etc. So, now the recovery and purification and purification of biosynthetic product from the growth medium is called downstream processing. Okay, so it is known as downstream processing. It includes technique techniques like filtration, centrifugation, sedimentation, next, and various types of various types of chromatographic techniques. Electrophoresis, etc. Next, the purified product is formulated and is mixed with suitable preservatives. So the purified product is formulated and is mixed with suitable preservatives. These formulations undergo through clinical trials. As in case of drugs, the purified product is formulated and is mixed with suitable preservatives. These formulations undergo through clinical trials, as in case of drugs. Downstream processing and strict quality control are essential before the product is marketed for human use. Okay. Now, next, now there is a last thing from this chapter is the applications of recombinant DNA technology. We know recombinant DNA technology has opened up wide opportunities for the service of mankind in various fields such as medicine, vaccines and growth of hormones, diagnosis, gene therapy, agriculture, improvement, and industrial fields. Okay, so now in my next video, I will discuss about these kinds of applications of recombinant DNA technology. So now, bye students.